Mechanism of action of phototropins. What are phototropins? How they act? What is their role in plants? Where they are synthesized? What activities they perform? Plants, unlike animals, are immobile. That is, plants cannot move; they are fixed. When plants are fixed and they cannot move, they would be having some mechanism to cope up with this limitation. That is, animals when see any danger, they can move from one place to another place. They can run away from fire. They can run away from any kind of wild danger. For example, human beings, when they see a wild animal, they run away. But plants cannot do it, and plants being immobile, but they would be having some other capabilities by which plants can cope up with this limitation. Plants thus are not like animals, and they respond to environmental stimuli differently from the animals. Animals have locomotion, animals have movement, and they can respond uh, the same way. For example, animals when see any kind of unfavorable conditions, they move away from those unfavorable conditions and return again when favorable conditions return. But plants cannot do it. Still, plants have the ability to adapt to the environment and to respond to different stimuli. Stimuli are any messages from the environment that trigger a response in the plants or living beings. One response in plants, one such response in plants is phototropism. What is this phototropism? The phototropism means, literal meaning of phototropism is, photo means light and tropism means movement. That is movement towards light. Movement towards light. And phototropism जो है, it is especially important for germinating seedlings. That is those seedlings which are yet to come out seeds from, which are yet to come out fully from seeds. For them phototropism is important, whereby the emerging shoot, that is the shoot which is contained in the germinating seed, that comes out, and this emerging shoot must grow. This must grow towards the light so that it can capture maximum of light for photosynthesis. Since this growing shoot requires light, requires food, and that food it has to prepare by photosynthesis, because now it has come out of the seed. Till now it was the embryo in the seed was nourished by the food reserves of seed. But however, when the seed germinates and shoot comes out, first radical comes out, then shoot comes out, and the shoot has to grow towards light so that it can do maximum photosynthesis, because in photosynthesis light is required, and which type of light is important for photosynthesis, and how plants respond to those light, how plants grow towards those light. We see the plants are positively phototropic, that is, plants grow towards light. Particularly shoots of plants grow towards light, while roots grow towards soil. That is, roots are positively geotropic and shoots are positively phototropic. Shoots are positively phototropic. Phototropins are actually, they are plant specific light activated receptor kinases. Plant specific light activated receptor kinases that is they are a kind of enzymes which are light activated, that is they require light for their activation. Yani in absence of light they cannot perform their function, that is they are activated by light, and they are plant specific, and they are receptor kinesis. Receptor kinesis means they are kind of receptors, like we have receptors on antennae, antennae as receptors for receiving the signals for receiving the signals for receiving the signals for our TV sets. So are these phototropins. They are plant specific light activated receptors, but they are a kind of enzymes. Which kind of enzymes? We call them kinases. Phototropins are thus receptor kinases which are activated by light. Kinases means enzymes which add phosphate groups or phosphorus, and these photoreceptors regulate they regulate diverse diverse conditions, diverse responses in plants. And what they do, they serve the plant to optimize the plant growth under various light conditions. That is, plants can grow fully under various light conditions by means of harvesting maximum light by these photoreceptors. So phototropins are a kind of photoreceptors. Their role is the mechanism of addition of phosphorus. That is why they are called kinases. Phototropins, point number second, phototropins undergo autophosphorylation. This is the mechanism of action of phototropins. That is, they undergo autophosphorylation as an essential step. They undergo photophosphorylation as an essential step. They undergo photophosphorylation as an essential step. This is autophosphorylation. Autophosphorylation means they undergo phosphorylation by themselves. That is, they add phosphate group. This autophosphorylation, this is an essential step for their signaling. That is for signaling. Signal means message. For messaging. For giving a message to the plant. For optimizing maximum light harvest, and for directing the plant to grow, and other specific tissue specific actions, they undergo autophosphorylation and induce a variety of tissue specific or organ specific responses. That is, those responses which are specific to a particular tissue. There are so many tissues in plants. We have cambium, we have apical meristem, we have parenchyma, we have collenchyma, and each tissue responds differently to the light by means of these phototropins, or organ specific. Yani flower responds one way, stem responds other way, leaf responds other way, and there are divergent mechanisms how these responses are created in plants by phototropins, and most of them are unknown. Like photoreceptors associated with our vision. This is an example from our system, human beings. That is, for vision, yani for seeing any body, we have photoreceptors in our eyes. Those receptors receive the light signals and elicit a response in our brain to respond that stimulus provided by the environment, and we see different kinds of objects. That is, photoreceptors are here involved for seeing or responding to different kinds of objects related to sight. So in plants, they too have, like our photoreceptors, they have photosensors, and these photosensors can detect presence, intensity, direction, and color of light. This is important. Yani the photosensors in plants, they have the ability to detect the presence, they can detect the intensity, light kitni shadid hai, how intense is light, and where from light comes, yani direction of light, whether it comes from east, west, right direction, left direction. And what is the color of light? Color means, in technical terms, wavelength of light. For example, we have ultraviolet light, we have visible spectrum. Visible spectrum has violet, indigo, blue, green, yellow, orange, vibgayar. Violet, indigo, blue, green, or vibgayar. Violet, indigo, blue, yellow, orange, red. Those are wavelengths, are different colors. And these photosensors, that is phototropins, they have the ability to detect the presence, to detect the intensity, to detect the direction and the color of light. And in turn, these phototropins, what they do, they utilize this information, that is whether light is present, what is the intensity of light, what is the direction of light, what is the color of light. These phototropins use this information and help the plant to respond this information by plant showing growth to these responses. That is, plant responds to the presence of light, to intensity of light, to direction of light, to color of light, by it is growth. There are 4 types of photoreceptors which have been identified in plants, and among them is a small family of proteins known as phototropins. This means that phototropins are actually proteins, a type of proteins which are activated specifically by 2 types of wavelengths. One is ultraviolet wavelength and the other is blue wavelength of light. That is, phototropins for their stimulation, for their activation, require ultraviolet light or blue light. And phototropins, they not only control the growth of plants, not only control the direction of growth, but they control a wide range of events in plants. Like they control phototropism. Phototropism means, photo means light and tropism means movement. Phototropism, development of chloroplast. Chloroplast develops and it has stages. First, some thylakoids are formed, then thylakoids get into compact form to form grana. This is the development of chloroplast, and for development of chloroplast, phototropins are involved. Orientation of chloroplast. Chloroplasts have to orient in the cells to absorb the maximum light for photosynthesis, and that orientation is triggered by phototropins, and they help in opening and closing of cytomata. Opening and closing of cytomata is an essential process for transpiration of plants. They help in positioning of leaves. Leaves have to orient themselves to the light, or they have to move away from the light, and in this orientation phototropins are involved. They help in heliotropism. Heliotropism means helios to sun, and tropism movement, that is tracking the movement of sun. This phenomenon was used by early people in deciding the time of the day. They were tracking the movement of sun by seeing different positions of sun, by tracking the different positions of sun, and they track the movement of sun. Phototropins, that is, they are involved in heliotropism. It is most likely that the phototropins, they generate a specific output after event of autophosphorylation. This is important. That is, when phototropins undergo autophosphorylation. Autophosphorylation means that they are phosphorylated by themselves, and when they are phosphorylated, they create an output. Like we give input to some machine and it gives us output. ट्रांसफार्मर को हम इनपुट देते हैं लो वोल्टेज, इट गिव्स हाई आउटपुट वोल्टेज. इसी तरह when these phototropins are autophosphorylated, they give an output. And what is that output? We have to know it. Lately it was found that it is the blue light receptor genes which are present in Arabidopsis thaliana. Arabidopsis is an experimental plant from which we have developed so many models in genetics, in plant physiology, etc. And it contains 2 phototropins, and that is this Arabidopsis, it has been found to contain 2 phototropins, and these phototropins are referred as phototropin first and phototropin second. The structure of these plant phototropins, they can be separated into two parts. It is important for knowing the function of any structure, of any body, of any instrument. We have to know its structure, because structure is directly related to function. In this case also, plant phototropins, they can be separated structurally into two parts, like every kind of protein. Yeah, most protein, they have one terminal, that is N-terminal, which is photosensory. Photosensory means it is elicited or activated by light, photosensory. And this N-terminal, it acts as receiving the input, यानी यहाँ से ये इनपुट लेता है. N-terminal photosensory input region, and it has C-terminal effector region or output region. For example, we draw a line from this side, right side, left side, there is N-terminal, and on the left side there is C-terminal. C-terminal is output terminal and N-terminal is input terminal. From N-terminal it receives light stimulus, and from C-terminal it gives response. That is, it is output terminal. And C-terminal contains classic serine threonine kinase motif. Kinases are enzymes which are responsible for adding phosphate groups. N-terminal region, that is, we now know that phototropins have N-terminal and C-terminal. The N-terminal region, it comprises so-called LOV domains. These are important. What are these LOV domains? We will learn it in next slide. And these LOV domains bind to vitamin B based flavin mononucleotide. And this flavin mononucleotide, it is a blue light absorbing chromophore. Chromophores are substances which can absorb light, and so is the FMN, that is, it absorbs blue light. Because I already told you that phototropins can absorb blue and ultraviolet light. That is, they require ultraviolet and blue light for their activation. And the part that is responsible for sensing that blue light or ultraviolet light is FMN. And this FMN is present at the N-terminal in the domains which are LOV domains. LOV domains, these, what are these? They exhibit protein sequence. यानी इनमें प्रोटीन सीक्वेंस होती है. They have arrangement of proteins, homology to motifs found in diverse range of eukaryotic and prokaryotic proteins. That is, they are similar to diverse range of eukaryotic and prokaryotic proteins which are involved in sensing light. That is, they have the ability to sense light. That is, they receive light, they respond to light. This means the sensing of light. And this LOV is called light, oxygen or voltage. So the acronym LOV, light, oxygen and voltage. We have a technique by which most proteins are studied. Their three-dimensional structures are studied. That is subject matter of proteomics. The technique is called protein crystallography. That is, crystals of proteins are prepared, and then the three-dimensional structure of proteins is viewed under high power microscopes, and the structure of proteins is modulated. And by this technique, that is protein crystallography, it has shown that LOV domain, it consists primarily of 5 antiparallel sheets and 2 helices. And these 5 antiparallel sheets and 2 helices, they bind to FMN. This FMN, that is flavin mononucleotide, is the actual site for absorption of blue light, and it has an enclosed structure. What is meant by this 5 antiparallel sheets? That is, for example, this is one sheet number 1. It has 2 terminals, N-terminal and C-terminal. The other sheet would have C-terminal and N-terminal, C-terminal and N-terminal, C-terminal and N-terminal. Likewise, the third sheet have N-terminal and C-terminal. This is called the antiparallel sheets, 5 antiparallel sheets. This is protein phototropin structure. The A structure gives the visualization of this structure of phototropin. We have two domains. This is at the end terminal. These are photosensory domains. That is, they can absorb the light and then can respond to it. This is that C terminal. This is the output domain. This is the output domain. It is rich in serine, threonine, amino acids. This is J alpha helix. All are shown here. And this LOV1 and LOV2, these are the domains that are linked to FMN. This is these three-ringed structure. This is FMN, flavin mononucleotide, and it has ability to sense blue or ultraviolet light. This is LOV domain at the extreme N terminal. Then we have after it LOV2 domain. This is J alpha helix. This is the overall structure of model of this phototropin. This is the output region. This is the input region, and this is the crystallographic structure observed under microscope. This is protein folded structure, and here it has FMN, that is this part. It has these LOV1 and LOV2 domains. Today, that is till now, it is still a mystery. It is a secret. It is not revealed yet. It is not known as to why phototropins contain two LOV photosensors. That is why there are two LOV photosensors. Why there is not only one LOV photosensor? Photochemically active LOV2 is, that is LOV1, not LOV1. It is LOV2 which is necessary for phototropin function. While the presence and photochemical reactivity of LOV1 has been shown to be not essential, then it becomes the challenge for biochemists to know why there is LOV1 when LOV1 is not essential for photosensation. It is only LOV2 which is essential, and it is a mystery and it is not known yet. However, LOV1 has been proposed a mediate receptor, that is, it is as a mediator, that is, it passes the information and it helps in dimerization. Dimerization means formation of two units, or it modulates the photochemical reactivity of LOV2, that is, it enhances the photochemical reactivity of LOV2, or it helps the LOV2 to sense better, to have good photosensory power. And LOV domains, when they are expressed, that is, they show their function, and when they were purified from bacterium Escherichia coli, E. coli, they are yellow in color, and owing to their bound flavin cofactor, that is FMN, flavin mononucleotide, they are photochemically active in solution as monitored by absorbance spectroscopy. We have this phenomena, absorbance spectroscopy, and by which we can know. We have spectrophotometers installed in sophisticated laboratories where we can see that whether the substance is photochemically active or not. It depends upon whether the substance absorbs in solution or not, and it has been found that LOV domains are yellow in color and they are photochemically active even in solution, that is in outside the system, and this has been proved by absorbance spectroscopy. In the dark, when there is no light, LOV domains bind to FMN, flavin mononucleotide, non-covalently, not by covalent bonds, not rigidly, not tightly, and they form a spectral species that is called LOV447, and absorbing, that is, this LOV447 absorbs maximum at 447 nanometer. Nanometer is the unit of light, wavelength of light, measured in nanometers. That is, it corresponds to violet region of the spectrum, visible spectrum. Irradiation of the domain, when light is made to fall on this LOV domain, but it induces the formation of a covalent bond. Then after, when light falls on the LOV domain, first FMN is not covalently bound to the LOV domain, but when light falls on the LOV domain, this covalent bond is formed between the C4A carbon of the FMN and the sulfur atom of nearby conserved cysteine amino acid residue within the LOV domain. And when formation of sterile adductor occurs within microseconds of illumination. That is, when illumination occurs, when light is fallen on this LOV domain, it happens in microseconds and it produces spectral species that is called LOV 390, and it absorbs maximally at 300 nanometer wavelength. And when LOV 390 is fully reversible in darkness, that is, it reverts back to its original form and represents the active signaling state. This is the actual signaling state. This is the actual state which is responsible for photoreceptor activation or the final process of phototropin. Phototropin, this is a bellulite receptor kinase. Again, it is a kinase, that is, phototropins are kinases, that is, they have the ability to absorb, they have the ability to add phosphate groups, they undergo autophosphorylation, and they are involved in bellulite responses of several species. Bellulite means from the visible spectrum we have 7 wavelengths: violet, indigo, blue, vibgire, blue, green, yellow, orange and red. And these phototropins, they usually respond to this blue wavelength of light, and phototropin converts blue light signals from the environment into biochemical signals. That is, a biochemical signal is created in the plant system, and as a response, cellular responses are created in the plant. Cellular responses. Phototropins are activated by light. How they are activated by light? As per the current knowledge, current view of phototropin receptor activation, that is how phototropins are activated, is that LOV2, it is the actual domain which is responsible for function of phototropins. It functions as a receptor of C-terminal kinase. C-terminal kinase, since phototropins have 2 terminals, one is N-terminal and the other is C-terminal. At the C-terminal acts as a kinase that is responsible for addition of phosphate group in the dog. This happens in dog, and that this mode of repression is elevated upon photoexcitation. That is, first, LOV2 functions as a repressor of C-terminal kinase. That is, LOV2 does not, LOV2 represses, or it hinders, or it stops the C-terminal from its activity of being an output, or being an kinase, or being an agent of autophosphorylation. That is, autophosphorylation does not occur in dog, but this is reversed. This repression is removed. This blockade is removed upon photoexcitation. That is, when light is made to fall on the phototropin. And what happens when light falls on the phototropin? Receptor, this phototropin receptor, autophosphorylation occurs. This is the main phenomena by which these phototropins act. Autophosphorylation. The photoexcitation of LOV2, that is when LOV2 is activated by light. This is called photoexcitation. That is, electrons move away from inner orbitals to outer orbitals, and what occurs is displacement of their junction J alpha helix from the surface of domain, and this helix unfolds, and this is designated as J helix, and by this C-terminal kinase domain is activated. C-terminal kinase. Protein rearrangements within the central scaffold of this LOV2 domain. That is, there occurs protein rearrangement. This brings about the protein changes at the surface, and these are necessary for activation of C-terminal kinase for autophosphorylation. That is, this is important. Autophosphorylation, the happening of autophosphorylation is important for phototropins to act, and this autophosphorylation is done by the C-terminal. So C-terminal kinase is actually the output side. When light activated phototropin can return to its non-phosphorylated state upon incubation in darkness. That is, when darkness comes again, the phototropin becomes again inactive. And how this deactivation occurs? Deactivation occurs by dephosphorylation. Activation occurs by phosphorylation. Phosphorylation means addition of phosphorus, and dephosphorylation means the removal of phosphorus. Yani by removal of phosphorus, phototropin becomes deactive, and by addition of phosphorus, phototropin becomes active. And the addition of phosphorus, phosphate group, occurs at the C-terminal, and so C-terminal C-terminal is the actual site for action of phototropism. This is the activation of phototropin. In this figure, this J alpha helix, this red part, that is C-terminal. This is bind to these two domains. That is, these two domains are repressed. They cannot do their action. They are folded. Like a human being, when has folded limbs, arms tied, he can do nothing. And when the arms are unfolded, they are untied, we can move our arms and do certain functions. This is the case here. When this C-terminal is folded back on these LOV2 and LOV1 domain, this is inactive form. And when blue light falls on it, this unfolds, and here autophosphorylation is occurred. This is phosphate group, this is phosphate group. This at C-terminal autophosphorylation has occurred. This is the active form of it. This is the active form of it. As is written here, photoexcitation of main light sensor LOV2 causes a disordering of the G alpha helix and activation of C-terminal kinase domain. That is, this is activated, which consequently leads to autophosphorylation of photoreceptor. Autophosphorylation at a conserved serine residue, that is an amino acid, which is labeled as SER, serine residue 851, within the kinase domain of Arabidopsis phototropin first. This is essential for photoreceptor signaling. And the sites for phototropin autophosphorylation have been mapped upstream of LOV2. There is still no information where these sites are actually located. And these are beyond your scope. This is important. If we see at this diagram, this is LOV1 domain, this is FMN attached to LOV domain, FMN thalamin mononucleotide attached to LOV2 domain. This is J alpha helix, and this kinase domain is folded on it, and blue light upon the falling of blue light on this phototropin. What happens? Autophosphorylation occurs. These three P's. This is the autophosphorylation here. These three P's. FMN becomes attached covalently to LOV domain. This again FMN. This line here, it shows attachment of FMN to LOV domain. Now this FMN is the actual site for absorption of this blue light. And what happens? This kinase domain, this gets unfolded from this N-terminal domain, that is the domain containing LOV1 and LOV2, and this kinase domain, it too is phosphorylated. This is called autophosphorylation, and this phosphate is provided on the substrate. The substrate would be contained in the cells for the elicit of responses. For example, phototropins elicit many responses. They induce many responses in plants, like opening and closing of stomata, orientation of chloroplast, development of chloroplast, phototropism, heliotropism, that is tracking of sun. For that, autophosphorylation is required, and that phosphorylation is provided by the this kinase part, kinase domain, C-terminal domain. This domain from this side, left side, it is the N domain, N-terminal, and this terminal kinase which is attached to this is the C-terminal. Again here it is N-terminal, it is C-terminal, but unfolded. This is the active form, this is the inactive form. This in darkness, what happens? These phosphates are removed, and this again becomes folded. This phosphorylation occurs, phosphates are removed, dephosphorylation occurs. This is activation by blue light, and this is deactivation by dark wavelength. Phototropin signaling, it is beyond your scope. It is not that much important, but actually phototropism ultimately results in an increase in growth on the shaded side of system. That is, that side which does not face the light, that side grows more than the side which is in direct contact with light. What happens actually, because on the shaded side oxygen accumulates, and oxygen hormone has a role. It is a plant growth regulator, plant growth substance. It helps in cell elongation and growth. So shaded side grows more than the side, illuminated side. So differential growth occurs, and by this differential growth the branch or stem bends towards light, because on shaded side when more growth occurs, the band is created, and that band will help the branch or the stem or the leaf to grow towards light. As light passes through the stem, it becomes progressively diffracted, thereby generating a gradient of phototropin activation across the organ. Highest level of activity of phototropin occurs on the irradiated side, that is on the side where phototropin is eliminated by light, and lesser activity is on that side where phototropin does not receive the light, and on the side from which phototropin is irradiated, more phototropin becomes more active, and more induction of response, more induction elicitation of response occurs. Formation of this biochemical gradient, that is photochemical gradient, biochemical gradient, that is on one side there is more activation of phototropin, on other side there is lesser activation of phototropin. This underlines the directionality of the phototropic response. That is why the plants respond to towards light. Presently, little information is available regarding how a differential stimulation of phototropin activity across the stem results in accumulation of oxygen on the shaded side. It is not clearly known, but likely affected the localization activity of specific proteins which are responsible for oxygen transport. That is, some proteins would be involved in oxygen transport, and so differential oxygen transport would be responsible for differential growth, that is more growth on shaded side than on the illuminated side. Phototropin signaling, that is how phototropin receives the light stimulus and then helps the plant to respond to this light stimulus. It is undoubtedly, that is without doubt, it is a very complex phenomenon, and many of the things are not yet known, and it elicits a wide range of responses in the plant. For example, it increases cytosolic, yani cytoplasm in the cytoplasm of cell. Calcium concentration is calcium in the calmodulin protein is a messenger of the cell, like we have messenger RNA. So is the calmodulin containing calcium, and these appear to be a signaling messenger downstream from receptor activation, particularly in rapid growth inhibition response. That is, plants do not show rapid growth inhibition, that is, rapid growth is inhibited. Plants do not grow rapidly, that rapid growth is inhibited when calcium accumulation occurs in cells, and this calcium accumulation is a response to phototropin signaling that occurs in seedlings emerging from the soil, that is in the seed germination. Besides cytoskeletal changes, there are cytoskeletal structures, cilia, flagella, microtubules, microfilaments in the cell. These are skeletal system of the cell. They are important for both belief-induced chloroplast movement, that is orientation of chloroplast, opening and closing of cytomata. But still, it is a major challenge for biochemists, plant physiologists to unravel how actually this signaling occurs. In darkness, phototropins are typically associated with plasma membrane, that is, they are bound to plasma membrane, but their small fraction is rapidly internalized, that is, they are internalized, they become free in the cytoplasm once the cell is illuminated. One consequence of autophosphorylation, this is the actual phenomena around which the whole mechanism of phototropins circulates, that is autophosphorylation. Autophosphorylation is the actual phenomena, that is, they add phosphate groups, and that addition of phosphate groups elicits the response. It creates the mechanism of action, and therefore to promote the receptor dissociation from plasma membrane and the desensitizing of photosensory system, analogous to other receptor kinase based signaling system. We have another consequence of phototropin autophosphorylation to mediate